In gene therapy research, lipid-based transfection agents such as lipofectamine are commonly used to deliver genetic material into cells. While effective, these commercial reagents can be expensive and are often derived from synthetic lipid formulations. In this innovation, we propose the use of avocado seed oil as a natural lipid source to develop a lipid nanocarrier system that can function as an alternative DNA delivery vehicle. Lipids extracted from avocado seed oil are formulated into nanoscale lipid nanoparticles. These particles form a lipid nanocarrier capable of transporting therapeutic genetic material. Therapeutic plasmid DNA carrying the CRISPR-Cas9 gene editing system is encapsulated within the lipid nanocarrier. The lipid layer protects the DNA from degradation within the biological environment. After administration, the lipid nanocarriers circulate through the bloodstream. Their nanoscale size allows efficient transport throughout the vascular system. As the nanoparticles travel through the body, they accumulate near breast cancer cells due to the abnormal vascular structure surrounding cancerous tissue. The avocado seed oil-based lipid nanocarrier interacts with the membrane of breast cancer cells through lipid membrane interactions. Following this interaction, the nanoparticle enters the breast cancer cell through endocytosis, a natural cellular uptake process. Inside the cell, the lipid nanocarrier disassembles and releases the therapeutic plasmid DNA into the cytoplasm. The DNA then travels to the nucleus, where the CRISPR-Cas9 system targets specific genes responsible for breast cancer cell growth. Through this gene editing mechanism, the proliferation of breast cancer cells can be inhibited, or the cells may undergo programmed cell death. Compared to conventional transfection agents such as lipofectamine, avocado seed oil offers several potential advantages. As a natural and renewable lipid source, it provides a more sustainable approach for developing lipid nanocarriers while also utilizing agricultural waste.
This research was conducted in several stages. First, avocado seed powder preparation was carried out by separating the seeds from their skins, washing, cutting them into small pieces, drying them under the sun for 3 days, and grinding them into powder, followed by sieving through a 50-mesh sieve to obtain uniform particles. Second, avocado seed oil extraction was performed using a Soxhlet apparatus. A total of 100 grams of avocado seed powder was extracted with 500 milliliters of 99% N-hexane at 100 to 110 degrees C for 5 to 7 hours to obtain pure oil for the formulation of GENOVA lipid nanoparticles. Third, plasmid preparation involved PM Stay Gold I3, which produces green fluorescence, and PX459V2.0-hypocas9M-Ruby2, which contains the hypocas9 gene used for CRISPR-Cas editing. Next, lipid carrier nanoparticles were produced by combining the isolated plasmids with avocado seed oil and other components using a mini extruder to form nano-sized lipid carriers. After that, physical properties were analyzed using a particle size analyzer to measure particle size, polydispersion index, and zeta potential. Finally, in vitro testing was conducted on MCF7 cancer cells to evaluate the performance of the formulation. By transforming avocado seed oil into a lipid nanocarrier system, this innovation introduces a promising and sustainable alternative for delivering gene editing therapeutics, specifically to breast cancer cells.
GENOVA is an innovative product based on avocado seed oil-based lipid nanoparticles, which contrasts with lipofectamine that utilizes synthetic cationic lipid formulations. In terms of ingredient benefits, GENOVA offers advantages as a plant-based, non-synthetic, and organic lipid source that can be developed into mass-produced lipids. Conversely, lipofectamine contains synthetic cationic lipids with high transfection efficiency, but carries the potential for side effects at certain concentrations. Regarding patient comfort, GENOVA has a lower potential risk for irritation and inflammation because it utilizes natural ingredients that are more biocompatible, making it a promising choice for therapeutic use beyond the laboratory. In contrast, lipofectamine is generally only applied for in vitro laboratory studies because it can cause cytotoxicity at high doses. Finally, from the aspect of environmental friendliness and sustainability, GENOVA supports a circular economy by using renewable and plant-based sources, thereby reducing dependence on industrial chemical-based synthetic lipids. On the other hand, lipofectamine is produced from synthetic chemicals and non-renewable sources, resulting in a manufacturing carbon footprint that is relatively higher.